I will focus today on desensitization of acid-sensing ion channels, and this will actually be a little bit of a trip down memory lane for me since it touches upon several projects of my postdoc, but all kind of through the lens of desensitization. So I will talk about how peptide binders and mutation bias desensitization and what structural insights reveal about the underlying mechanisms. But let's start with the basics. So acid-sensing ion channels, or ASICs, are trimeric channels that primarily conduct sodium ions. Each subunit contributes a large extracellular domain that is sensitive to changes in protons, meaning that decreasing the extracellular pH leads to the protonation of specific residues in the extracellular domain. And these protonations then trigger conformational changes that eventually activate the channel and open the pore. So this process is pH dependent with an EC50 of around pH 6.6. Today, I will only be talking about homomeric ASIC1A, even if I don't mention it specifically. They are highly expressed in the brain as well as in the peripheral nervous system. And under physiological conditions, extracellular pH is usually tightly buffered, right? But in the synaptic cleft, for example, pH can transiently drop to levels sufficient to activate postsynaptic ASIC1A, and this then contributes to synaptic transmission. Consistent with this, knockout of ASIC1A in rodents has shown to alter learning and memory with a particular impact on fear-related behaviors. In addition, there are several pathological conditions, including inflammatory pain, stroke, and cancer, that are associated with hypoxia-induced tissue acidosis. And the evidence linking ASIC1A activation to these diseases starts to become increasingly strong, and especially in recent years. And importantly, this kind of growing mechanistic understanding has begun to really translate into some promising advances in drug development, with multiple approaches now emerging to modulate ASIC activity. However, despite this process, we still don't have ASIC-specific drugs on the market yet. And there is a continuous need to better understand some of the basic mechanistic properties of ASIC in order to target it pharmacologically. So functionally, ASIC1A produces a rapid transient current in response to extracellular acidification. And then this fast activation is followed by also fast and complete desensitization within milliseconds to seconds. I have already shown you the pH-dependent activation, but ASIC also undergoes something that we call steady-state desensitization. In this case, the sustained exposure to kind of mildly acidic pH does not activate the channel directly, but it instead reduces its availability for subsequent activation by strong acidification. And like activation, steady-state desensitization is itself pH dependent. So as the preconditioning pH decreases, the amplitude of the subsequent current also decreases, giving rise to this kind of not only to the activation curve that is pH dependent, but also to this steady-state desensitization curve. From a pharmacological perspective, this opens multiple potential strategies to inhibit ASIC. One approach is, of course, to directly inhibit activation by shifting the activation curve to more acidic pH values. Alternatively, one could also selectively actually enhance steady-state desensitization, and then this would also reduce ASIC activation. And this latter strategy might actually be particularly attractive under physiological conditions where extracellular pH often remains mildly acidic for extended periods of time. Furthermore, many peptide binders that we know of today affect steady-state desensitization, and we thus really want to get a better understanding of the underlying mechanisms. The structural data of EpIC suggests that protonation of these acidic residues in the extracellular domain leads to a more kind of compact extracellular domain. And these conformational changes then promote straightening of the transmembrane helices and opening of the pore. Now, during desensitization, the upper ECD remains in this kind of protonated compact state because we're still under acidic conditions. But then the lower part of the channel kind of relaxes more towards a resting-like conformation. And this almost hybrid conformation builds the desensitized state. That said, how these states are actually exactly connected during gating is still a little bit debated, and it remains a bit unclear whether a single desensitized structure can account for both acute and steady-state desensitization. And this is exactly something we would like to explore. So the rest of the talk revolves around three questions. How does a known modulator affect steady-state desensitization? How can we use mutation to dissect the mechanisms of desensitization? And what structural data can we use to reveal some of the desensitized states? I will first begin with looking at a binder, a very potent binder called psalmotoxin, here shown in teal. It's a 4D residue peptide isolated from tarantula venom. And structural studies have shown that it binds at the subunit interfaces of two neighboring acid 1A subunits. And quite extensive animal work has demonstrated that PCTX can reduce infarct volumes, and it also improves outcomes in rodent models of stroke. Functionally, PCTX is remarkably potent inhibitor of EpIC1A. It effectively induces steady-state desensitization even at physiological pH, leading to nanomolar inhibition of channel activation. Because of this potency, PCTX is widely used in the EpIC field and often serves kind of as a benchmark control when testing for new modulators. It is therefore really critical to understand its mechanism of action in detail. And this is already where it gets a little bit more complicated because PCTX does not simply shift steady-state desensitization, but it actually acts as a gating modifier. So it shifts both the steady-state desensitization, but also the activation curve. And this then kind of raises several questions. So, for example, what are the conformational consequences of PCTX binding? And does it kind of bind in the same way at different pH values? To address these questions, we used voltage clamp fluorometry. And for this, we introduced a cysteine mutation at the top of the extracellular domain, in a position that has previously been found suitable for this purpose by the Stefan Kellenberger's group. And then we expressed the channel in Xenopus oocytes and labeled it with an environmentally sensitive fluorophore that changes in fluorescence intensity when the channel undergoes conformational changes. So then we record simultaneously the ionic currents that report on the functional state of the pore. And we also look at the fluorescent signal, which serves kind of as a proxy for the conformational changes that the extracellular domain undergoes. And as you can see, upon acidification, we start to observe fluorescent changes in the extracellular domain much earlier than we actually see activation of the channel. And kind of indicating that some of these conformational changes happen way before the channel actually opens. If we then kind of plot this, or inversely plot these fluorescent changes and lay it on top of a steady-state desensitization curve, we can see that they really nicely overlap. And this indicates that by looking at the changes in fluorescence, we effectively track the channel entering steady-state desensitization. So we thought this would be a very useful tool to then examine the effects of psalmotoxin. And when we look at the functional data of the pore, we see that psalmotoxin inhibits ASIC activation. From the fluorescent data, we see that the channel undergoes a conformational change that is somewhat similar to that one induced by these moderately low pHs, both in direction, but also in this kind of in the time course. However, in contrast to the functional recovery from it, we see that conformationally it remains in this PCTX-induced state. And when we record this a little bit differently and activate the channel several times, we can see that functionally the channel really nicely recovers, but the conformational signal persists, indicating that PCTX stabilizes this kind of desensitized like extracellular conformation that outlasts its functional inhibition. And only switching to really high pH, so pH 8.4, leads to an immediate return of the fluorescent signal to baseline. However, it's kind of, as soon as we step back to pH 7.4, it's like the channel remembers and goes back to this long-lived PCTX-induced state. And we can also record this a little bit differently with longer exposure to 8.4. But again, it's really like the channel seems to remember PCTX for a really long time. Given the stability of this PCTX-induced conformation, we kind of wondered how relevant it actually is and whether this state could influence binding of other ASIC modulators. So to test this, we turned to big dynorphin. And big dynorphin is the most potent known endogenous ASIC modulator. It is a neuropeptide which competes with PCTX for binding and induces a distinct closed state. In our experiments, big dynorphin also produces this kind of characteristic downward deflection of the fluorescent signal. And strikingly, when we pre-expose the channel with PCTX and then apply big dynorphin again, then the changes that we see are much less prominent than if the channel hadn't seen PCTX before. So although this PCTX-induced conformational state appears to be decoupled from pore function, it directly alters binding and action of other binders of the extracellular domain. So this is therefore an important observation and needs to be taken into consideration when searching and developing for other ASIC modulators. Without going too much into detail, we were able to kind of disentangle at least 3 distinct binding modes for PCTX. One of those was stabilized this long-lived extracellular conformational state that persisted at physiological pH and functionally kind of reshapes the binding landscape for other ASIC modulators. We also saw that PCTX conditions have profound influence on, we also saw that pH conditions have really profound influence on how PCTX modulates ASIC and affects both activation and steady-state desensitization. And together, kind of these results show that PCTX provides a really strong control over desensitization, but it is somewhat imperfect since it affects both since the effects are quite broad, they're state coupled and really highly pH context dependent. So this raised kind of a question for us. Can we tune desensitization more precisely in a different way? And to address this, we kind of turned to the protein itself and asked whether individual residues could shape desensitization more selectively. And one residue that has been implicated in at least acute desensitization of EpIC is a conserved lysine at position 211 up here. And previous work has shown that mutation of this K211 site dramatically accelerates desensitization kinetics, and it also shifts the pH sensitivity quite dramatically to lower pHs. To gain deeper mechanistic understandings, we systematically modified the side chain of this position 211 using non-canonical lysine analogs, allowing us to kind of generate side chains that are shorter, longer, or uncharged. And to incorporate these lysine analogs, we used a semi-synthetic approach. So the region containing this 211 position and its neighboring residues were synthesized by peptide chemists, while the remainder of the channel was expressed heterologously in xenopusoocytes. And then we attached split intine pairs on all fragments, and this allowed us then to kind of have a channel that has now kind of a scarless integration of the synthetic peptide and the intine pairs to splice themselves out. And this leads then to a functional channel. This approach allowed us to then kind of really look at near atomistic precision, what properties of K211 are critical for desensitization. So we first examined channel activation, and we've seen that shortening of the side chain leads to a slight loss of pH sensitivity, but really making the side chain longer or uncharged had dramatic effects on pH sensitivity. And consistent with this, all lysine analogs actually accelerated the desensitization kinetics compared to the wild type here in black. So now that we knew it affects this kind of acute desensitization, we of course wanted to know if it also affects steady state desensitization. And surprisingly, it did not. Despite the dramatic changes that we've seen on kinetics, it had really not really a measurable effect on steady state desensitization. And this, in a way, tells us that this K211 site plays a role in how stable the open state is, kind of shapes how the channel leaves the open state into desensitization, but it does not really control steady state desensitization. So to probe this mechanism a bit further, our collaborator, Joseph Harrison, ran some molecular dynamic simulations, and these revealed that only the native lysine side chain, but not the shorter, longer, or uncharged analogs could efficiently coordinate a chloride ion in the open state. And guided by this, we returned to the functional data and tested whether chloride availability actually affects desensitization kinetics. So when we reduced extracellular chloride, the wild type channel then showed accelerated desensitization kinetics, consistent with the notion that the chloride indeed is needed to stabilize the channel in the open state. And if it's missing, the open state is destabilized and the channel does desensitize quicker. In contrast, in channels that contain the lysine analogs, they are already unable to coordinate the chloride ion effectively. So then removing it doesn't really lead to a further change in kinetics. And this supports a model in which K211 very selectively stabilizes the open state through chloride coordination without contributing to steady state desensitization. It also, of course, then raises the question, can we find another residue that controls desensitization a bit more directly? And for that, we then turn to the nearby residue N414, which is located in the kind of for EpIC famous beta 1112 linker. And this linker has been extensively studied, particularly by work from David McLean's lab, and is well known for its central role in desensitization. And multiple studies have shown that flipping of this beta 1112 linker is tightly coupled to the rate of desensitization. And mutation in these regions can dramatically alter the speed of acute desensitization. So, of course, the first thing we wanted to look at was steady state desensitization and looking at it using the fluorescence approach that I already talked about before. And just a quick reminder, in the labeled wild type, the channels closely the fluorescence that we see closely tracks steady state desensitization, indicating that the fluorescence reliably reflects entry into the steady state desensitized state. And in this N414K mutant, however, we not only see now much slower acute desensitization, but really also no steady state desensitization anymore. So we don't have this kind of nice inhibitory curve anymore. But if we look at the conformational changes, they are basically untouched by the mutation and still so this extracellular domain still very much is sensitive to pH. It's just that the mutation seems to have decoupled the conformational changes from the functional changes in the pore. We've extended this analysis to several additional labeling positions across the extracellular domain, and without, again, going into too much detail, the labeled wild types all kind of in all positions closely tracked, the fluorescence closely tracked steady state desensitization. However, when we introduced the N414K mutation, a quite persistent picture emerged. So functionally, these channels show little or no steady state desensitization, yet the fluorescence change remains intact across the extracellular domain. In other words, so the extracellular domain adopts a conformation that looks like desensitization, but in the presence of the mutation, these conformational changes are not longer coupled to the functional desensitization of the pore. And this uncoupling was not limited to acute desensitization, steady state desensitization, but it also extended to a third form of desensitization that I haven't talked about yet. This form is a phenomenon referred to as tachyphylaxis. It has been observed for wild-type EpIC currents, even in native tissue, and describes kind of a progressive loss of response upon repeated activation. Tachyphylaxis is thought to be linked to proton influx and subsequent changes in the intracellular domain of the channel. And in wild type, the effect is typically quite modest. However, in this N414K mutant, we see a strong use-dependent loss of current. And when we then examine the conformational changes via the fluorescence, we can see that the fluorescent changes remain very stable across the activations, indicating again that the extracellular domain is perfectly able to sense pH. It's just that the mutation uncouples the extracellular domain from functional desensitization in the pore. Across acute desensitization, steady state desensitization, and tachyphylaxis, we see similar extracellular motions, but actually all have very different functional outcomes. And based on this data, we began to kind of suspect that desensitization is not a single uniform state at the level of the pore. And before kind of turning to some structures, I want to briefly show some more functional data that supports the idea that EpIC can occupy multiple desensitized states. Here we examined recovery from desensitization using whole cell patch clamp recordings of EpIC1A. And in these protocols, the channels were first allowed to desensitize, and then there was a very brief, so like 30 milliseconds exposure to high pH before the channel was activated again. And if we only let the channel kind of just desensitize, then the recovery is actually quite good. But if we let the channel desensitize for much, much longer, and then again have this very, very brief switch to pH 8, the channels can't activate anymore afterwards. and need a much longer time until they actually fully recover. So much longer is still quite small. It's like 5 seconds. So this time dependence of recovery indicates that prolonged desensitization drives the channel into a deeper desensitized state, out of which resensitization needs much more time. And really together, these observations kind of support the idea that EpIC does not enter a single desensitized state, but rather multiple desensitized states with distinct recovery properties. And it was actually excellent that somewhere along the way, we were approached by Isabelle Bakongi and her team to provide some input on some cryo-EM structures that they had of human ESIC1A. And in total, they actually resolved 11 new structures of human ESIC1A in detergent. 8 structures were from 4 different pH conditions, 2 in the presence of the peptide toxin METX, and 1 was from a mutant. And importantly, all structures were actually collected under identical buffer conditions with pH as the only variable. And again, I won't go through all of these in detail, but generally, these 11 structures fell into 6 distinct categories. First is kind of a high pH structure with domain swapped TMDs. What I mean by this is, we can see a little bit better in here. So here we have an extracellular domain that is deprotonated and thus kind of expanded. And the transmembrane domain has, especially TM2, has a break and kind of swaps over to interact with a neighboring domain. And we also see a re-entrant loop right here, kind of nestled between TM1 and TM2. The pore shows some clear constrictions, and overall, this state strongly resembles this of a previously determined resting state. So then we saw some more confirmations. The next one shows we saw at pH 7.5 as well as pH 6.5, where the channel adopts a fairly similar conformation as observed when we resolved it together with METX peptides. In this state, the extracellular domain of the channel actually adopts a compact protonated conformation, while the transmembrane helices are linear. And since METX can open the channel even at really like high pHs, as well as out of desensitization, we actually suspect that what we see here is consistent with an open state. So third, we also observed a state at pH 7.5 that resembled that one of a highly pH-sensitive mutant. This channel has, this mutant has the mutation is on the intracellular side of the channel, and functionally the mutant shows a stabilization of the open state, so with higher pH sensitivity in general and a reduced acute desensitization. In these structures, the extracellular domain is actually deprotonated, like in the resting state, but the transmembrane domains are now not domain swapped, but actually linear. And we think that this could actually be something like a pre-open state. And then kind of what we have all been waiting for is the structures that we resolved at low pH, so pH 5.7. Under these conditions, all structures share kind of a collapsed extracellular domain and a disengaged beta 11-12 linker. So here we again have like the N414 mutation or residue that I talked about before. Now it's N416 because we switched species from mouse to human. But again, it's this very important linker. And when it's flipped, it's a telltale sign of a desensitized channel. So here we are looking at desensitized channels. And again, they all share very similar conformation in the extracellular domain, but the transmembrane domain adopts some really striking different conformations. We have a structure where the transmembrane domain adopts kind of the swapped conformation that we see in the resting state. And as a whole, this is actually very similar to previously reported desensitized channels. But we also have a structure with straight helices and one where we kind of have features of both. And all of these features that are really well positioned to influence ion permeation and, of course, also recovery kinetics. So this was actually quite unexpected, and it suggests that functional desensitization might indeed not be represented by a single structural state at the level of the pore. And at this stage, we really don't know yet which specific functional desensitized state these structures really correspond to. However, the fact that we do observe such structural diversity within what we kind of functionally call desensitization in itself, I think is really encouraging. And of course, in the future, we would like to establish more direct structure-function relationship for these desensitized states. And kind of the final figure of the paper then also summarizes the idea of kind of a simple model where closed states have a deprotonated extracellular domain, and protonation seems to indeed kind of lead to a straightening of the transmembrane helices into an open state. And then rather than entering a single desensitized endpoint, EpICs can, the desensitization can occur in multiple desensitized states that share similar extracellular domains, but really differ at the level of the pore. And these states likely differ in stability and recovery kinetics, and providing a structural basis for the functional diversity we observe during prolonged or repeated activation. So taken together, our work shows that desensitization in EpIC1a is likely not a single endpoint, but more something like a landscape of states with distinct structural and functional properties. Mutations can either have very specific effects on the type of desensitization, like in the case of K211, or it can be more broad effects that affect desensitization in general, like it was the case for the N414 residue. During the different types of desensitization, it actually seems like that the extracellular domains remain similar, but produce distinct pore states. And the conformational diversity that we saw in the structures could actually really provide new opportunities for selective targeting of EpIC in desensitization, and thus kind of promises new strategies for therapeutic targeting of EpICs in disease. With that, I want to thank first Stefan Pless for not only being a really great supervisor during these projects, but also actually a super mentor while taking this step towards a more independent position. And then some people I want to mention. So for the PCTX project, it was mainly Christian and Jene who were driving it. And then for the project with the lysine analogs, it was Debayan and Jacobo that drove the project, and Sophie, Gül, and Joe that contributed to it. In the slow desensitization project, it was my master students, Caroline, Asli, and Jos, that are now all PhD students. And for the structure function project, I want to thank Isabel and her team that was led by Kim and James. And then from our side, Nadine and Mette that contributed some patching data. And then I also want to thank my new environment here at Linköping University for welcoming me and making this transition really nice. I'm currently not hiring, but maybe at the end of the year again. So just reach out if you are interested. That's it from my side, and I'm happy to take questions.
Brilliant. Thank you very much, Stephanie. That's a great tour of a new family of channels for the series. So that was a great introduction. We've got a number of comments and questions on the Q&A. I encourage everyone to add any more. The first is a comment from David Colhoun, who mentions that you couldn't distinguish the models that you showed early on, your different kinetic schemes, with macroscopic methods. Only single channels could do that. Has there been single channel analysis of these ASICs?
Actually, not that I know of. I feel like Dave McLean would know this better. But the modeling so far that I'm aware of is actually really done mostly on yeah, not on patch data, not on single channels. And yeah, yeah, I'll leave it there.
And then a question from Esteban Suarez Delgado. Hi, thanks for this interesting talk. Have you measured the unbinding kinetics of PCTX1 with voltage clamp fluorimetry?
So no, we haven't. There was some FPR measurements done by others who saw that it actually seems to bind for a really, really long time. At that time, that was published earlier, and I think people didn't quite believe it. And now, or also we didn't know how relevant that is, right? And I think now this data kind of supports that the molecule seems to stay bound, kind of loosely bound for quite some time.
Damien Bell asks, with the various cysteine substitutions for Alexa 488 labeling and structure function studies, did you try any MTS reagents to chemically switch the cysteines to positive, negative,
Uh,
neutral?
oddly, I actually haven't, but I know that some other people in the lab before me have actually done that, and um, I would say for many positions or for some positions, similar results, and sometimes um, uh, it was the results were not so clear, but hard to tell whether it was kind of the choice of labeling and reagent or just kind of the conditions of recording.
Another comment from David Colhoun says desensitization is also still largely obscure for nicotinic acetylcholine and glycine receptor channels, but isn't very important for either of them in real life. So maybe just pointing out the complexity of desensitization processes across these ligand-gated receptors.
Yeah, I think like many are interested in it. Many are kind of maybe struggling with it, and yeah, this is not an isolated problem, which is actually quite nice. Yeah, but I'm not sure.
A
I'm
question.
actually I
Sorry.
wonder how much we could learn like from how um, I think actually like some lessons we can actually learn across different ion channel types. Yeah.
How brief are the pH changes that take place in the synaptic cleft upon neurotransmitter release? In other words, how much desensitization of ASIC actually takes place during synaptic transition? Aren't the pH change pulses too brief for desensitization to occur to a significant extent?
Yes, that is a very good question. So, and there is a paper on it that I think it was from Canessa's lab. I'm not quite sure. But yeah, there is a paper on it, and basically, it's absolutely true. So for under physiological conditions, these um, pH changes in the synaptic cleft are really brief. I don't really want to state the number, but they're so brief that the that desensitization might not really play a role for synaptic transmission that ASIC contributes to. But then, of course, we don't. It's not just about synaptic transmission, right? It's also under disease or like at least acidic conditions during disease or disorders where suddenly these these pH changes are much more long-lived. But yes, I think when it comes to the synaptic cleft, that's a very good comment and absolutely true.
Thanks. Next question. Has work been done on mutation of the peptide toxin? And I think that question came in when you were talking about the PCTX1, not the later toxin. But I guess the question still stands for the structural studies as well, the toxin used there.
Yes. Let me think. So I think there is actually mostly work being done on the other side, and I didn't present any of it to kind of show the residues it interacts with in ASIC. And then we've done some work on big dynorphin, where we actually so kind of this peptide that binds in the same place as PCTX, and we've done some mutations in big dynorphin and could actually make it to a much less efficient binder. In PCTX, I'm actually unsure. I want to say I don't think so, but then I also don't want to discredit anyone's work. Sorry.
Next question. Thank you for the great talk. Is the modulation of ASIC by dynorphin physiologically relevant or visible?
Sorry, I missed the beginning of the question.
Is the modulation of ASIC by dynorphin physiologically relevant?
Um, Great question. So um, and I think the shorter answer is we don't know yet. In cultured neurons, exposure to big dynorphin is actually so big dynorphin prevents steady-state desensitization. So we have more activation of ASIC, and that then leads to more cell death. But that's called, cultured neurons. Um, and so big dynorphin also binds to the opioid receptors, and probably the main physiological effects is through opioid receptors. How much of it also affects ASIC, I think we just don't know. But it seems like, kind of preventing that interaction could indeed be an interesting way of targeting ASIC.
A follow-up comment from Damien Bell who says, nice talk, thanks. Good luck, Dr. Heusser, in the hotbed of ion channels at LIU. And he attributes that. I get that copyright, so I'll work out the licensing agreement with the Chancellor. Next comment is from Mark Rogers, who says, lovely talk. Are these structure function insights translatable to other ASIC isoforms, such as ASIC-free? And are you planning to see if they are the same or changed in ASIC-1A, B, or ASIC-free heteromers? Which are known to occur in native cell membranes.
Yeah, I mean, simple answer, right? Like we don't know. I guess we would, in a way, expect to see similar transitions. I think, as you know, like the very specific conformations, they might actually differ from channel subtype and also like species. We've seen like some variations. So far, we just don't have the actual data for it. My guess would be that at least the concepts of it are translatable within different subtypes, and then that kind of the exact residues and that kind of makes up how, right? It's kind of like we see it in function, how quickly the desensitized state, or how quickly the channel falls into a desensitized state or can come out of it. But I would expect them to be similar, but I'm looking forward to actually seeing it.
Are those key residues conserved across the different family members?
Yes. So for example, like the beta 11-12 linker is very conserved among different species and different subtypes. Yes. Yeah. I think we still don't, right? We still don't quite know what some of the most important residues would actually be. But yes, so there is some of them are really, really highly conserved.
And David McLean says, great talk. The major differences of DLDDS or RRDS structures seem to be all at the level of the pore, yet the fluorimetry is measuring local changes near to the top or middle. Do you see large structural differences between desensitized states in the extracellular domains that might hint at where the VCF changes arise?
Yeah. So let me just go back to actually show that. So just so people kind of know what he was talking about. Yes. So basically, all structures show a similar ECD, and then we see changes here in the TMD, and kind of the labeling that we've been doing is some of it happens really at the top of the TM1 and TM2. So I kind of changed the way I've been thinking about it because in a way, what we see with the fluorescence is quite similar. Uh, I don't know, similar. Let me see if I can find where. So quite similar effects on the fluorescence, no matter whether it's for I don't know. Let me whether it's kind of desensitization, acute desensitization. So if you would let this go on for a little bit longer, or if we steady state desensitize, and then also if we have tachyphylaxis, actually like the fluorescent signal are all also quite similar, just like in the structures, right? So maybe it's actually kind of the other way around that the with the fluorescence, it's easier to kind of really follow what function the or like yeah the type of desensitization that we are looking at, but actually it's not. I don't know if that makes sense, but the fluorescent signal is not so different between the different types of desensitization in the end. When it comes to some of the residues right here, and I haven't really shown all of it because it gets a bit complicated, we actually start to see we can with the fluorescence we can actually follow the steady state desensitization, but we can also follow activation, and the signals kind of get mixed up, and it gets really difficult to kind of dissect them from each other. But that kind of going back to the structures, I think what we see is that the top, so the top of the TMDs are quite similar, but slight. You really need to look at the structures. Slight variations that, of course, we could be able to pick up with fluorometry, but I think it's really difficult to link still our fluorometric data to the exact structural data, and we still struggle with it. Sorry, that was a bit of a jumbled answer. It's
No, I think that's good. And I think that kind of addresses a later question, which says that changes in fluorescence from a single site are used to infer the functional state of the extracellular domain. Couldn't such a simple probe be misleading when it comes to inferring something as global as the functional state of a whole domain, which I think is what you're saying in your answer there, right?
Yeah, yeah, and I
Sure.
think that's true. I think we do have to be always careful when interpreting these results. And I think we start to get more confident once either we see something across like the whole extracellular domain, or if we see it again and again under different circumstances. So, for example, for us, if we now see, right, we can follow different types of desensitization, and we can see how like changes in modulation that actually make sense, both like also on a functional level.
Excellent. Okay, we've got a couple of minutes before we're at the hour. Two questions from Peter Segura. You mentioned that the desensitization occurs in milliseconds. I'm curious about how the application of the ligand, protons in this case, in the oocytes, how long this takes, practically, I think, and how long does the recovery take?
Yes. So I wanna be so this depends. So of course in oocytes everything takes a bit longer. So it takes, less than a second, of course, to activate, and then it takes a couple of seconds. It depends a little bit on the species that you work with. So like, yeah, humans are a little bit, human ASIC is a little bit faster than mouse, for example, but it takes a couple of seconds. And that's fine, but it is like, it's fine to look at, pH dependence of activation in steady state desensitization, but especially desensitization, we are limited by the perfusion speed. So what we see is to some extent perfusion speed, absolutely, when we talk about data on oocytes. So this is why when we work with oocytes, the only thing that we might do is kind of like compare the desensitization rates, but it is limited by perfusion, absolutely. And then with patching, I think there we can be much more confident.
Great, and that covers a suggestion from David Colhoun. He said you might need outside out patch to look at rates, which I think is what you're getting at.
Yeah.
Final question then. In the context of transient acidification at the synapses or in ischemia, does your kinetics predict that the channel contributes more by the fast peak or to integrated signals driven by repeated events?
Okay. I hope I understand this question correctly. So is this and I'm not quite sure.
Uh-huh.
So
I guess it is the channel opening by an initial peak, and that's what's biologically meaningful, or is it continuously opening during sustained acidification?
Yeah, no, I think it is really like it has a peak and that is relevant.
Okay.
Yeah.
And maybe you can follow that up
And then
afterwards
it
because we should.
desensitizes fairly quickly, right? Under physiological conditions where everything goes correctly, the channel desensitizes.
Brilliant. Okay, well, thank you very much, Stephanie. That was an action-packed Q&A session. I guess if anyone has any follow-ups, they can reach you via email, right?
Absolutely, yeah, sure. Just reach out.
But we should let you go. It's dinner time. Thank you so much for joining us. We hope to see as many of you as possible in person this month. We've got comments coming in saying great talk. For those of you who can't join us in person, we look forward to seeing you here next month. So thank you everyone for joining, and thanks again, Stephanie. Well done.
Thank you, everyone.